Здравствуйте, меня хорошо слышно, видно?
Mark, hello.
Hello, меня хорошо слышно, видно?
Just a moment. Our administrator will organize a special session for you, because this one session is for our open winter TISP school. Okay.
Okay, thank you.
Потише, ребят. Добрый день. Слышно нас?
Да, вас хорошо слышно.
Да, здравствуйте. Надеюсь, меня тоже хорошо слышно.
Да, вам камера нужна, чтобы зал видел? Детей?
Мне скорее нужна доступ, чтобы видеть презентацию, если она есть.
Нет, есть.
Ну все.
Дети поделиться рабочим столом и дети начнут?
Да, можете через рабочий стол.
Все, хорошо. Все, ребят, что? Как вы ждут вашу?
Я уже отправила.
Отправили туда? Какую?
Hello, dear colleagues. Hello, participants of free online winter school of Tomsk International Science Program. Do you hear me? Is it okay with our internet connection?
Yes, I can hear you. Hello.
Hello, Nida. Thank you very much. Glad to see you. So, let me start our free online winter school, Tomsk International Science Program. Very glad to see you participants. But I hope that some participants will join us a bit later, because we received a lot of applications, and we try to answer for all of them and send to all of them the link for connection. So I suppose the number of today's participants will increase So I have the 10 minutes for introduction you to the Tomsk International Science Program. First of all, my name is Gulnara Voronova. I am a Deputy Director of Smart Materials and Technologies Institute at Tomsk State University. Also, I am a lecturer at Tomsk International Science Program. First of all, I'd like to share with you the information what Tomsk International Science Program is, because we organize a winter school for our future abiturients, for our future freshmen, and that is why it's very important to know what we are waiting as a result of winter school. We would like to see you among our students in 2022 years, or maybe later. So let me show you the short presentation. Do you see it? Thank you. While we are waiting the joining of our participants, I will introduce you what Tomsk International Science Program is. This is the bachelor degree program in Informatics, in the field of Informatics at National Research Tomsk State University. The head of program is a professor, doctor in chemistry, Irina Kurzina, but today she is on a business trip and she asked me to be a presenter of Tomsk International Science Program for you. Some keynotes about our programs. The field of study, as I said, is an innovation, and common duration of state study for bachelor program 4 years. It's a full-time program mode, fully taught in English. That's a bit rare for Russia education, so we attract foreign students, and now we have at our program students from all over the world, from Cameroon, from Italy, from China, from Indonesia, and so on, and also from Russia, of course. You see the tuition fee for program is a common level of fee for program in English, among others program at Tomsk State University. And if our abiturients show the excellent results at entrance exams, they can get the tuition fee discount. We have a lot of partners, but main of them are Chimie Paritech in Paris, France, and Siberian State Medical University in Tomsk in Russia. Why we start our program in 2019? So it's a new program, experimental program, even for our university, because our global society facing complex challenges, where many of challenges are interdisciplinary, which require a new generation of specialists, of scientists who are capable of looking across traditional boundaries between the traditional disciplines, traditional courses. And also we follow the internalization of science as a tendency at Russia, in Russia, and at Tomsk State University. And we hope that our students in the future can apply for the master program or can continue their career in a business or in any sphere. Our program is unique in Russia with a multidisciplinary approach in sciences, including chemistry, biology, physics, ecology. For every student, we offer a tailor-made program, which allow to emphasize the talents and focus on personal ambitions and interests of students. And our program organized in a liberal arts and science approach, where education is a central tool in the personal discovery and development of students. So some information about our program. Now we have about 30% of international students, I mean the foreign students. The applicants should pass some, of course, 2 exams for foreign. The main is a mathematics, and for choice, physics, chemistry, or informatics. You can try our demo version of every exams, and admissions for our program has already started. Some details about curriculum. The curriculum for Tomsk International Science Program students include courses on biology, chemistry, physics, mathematics, neuroscience from our partners, Siberian State Medical University. And of course, it includes mandatory courses and courses on choice. And our students have a lot of practice and training. Here you see the short demonstration how the education organized year by year from the first year to the end of program. You see the first year students have majority of the courses are obligatory. It's a core courses, and beginning from the second year, continue to the third year, students can choose some program and construct some courses and construct their own education track. It can be as a pure track in chemistry, or but we try to motivate students to choose the multidisciplinary track, which includes the disciplines from different tracks. Our students study in a PBL approach and also in a research-based learning approach. What we are waiting from our students after they end our program and award a degree of Bachelor in Innovation. We can offer for students the master programs as in Tomsk State University, and they can choose their own education track in other universities. In Tomsk State University, our students can continue study in biophotonics master program, biodiversity master program, or for instance, very popular for students, the master program, translation, chemical and biomedical technology. Or even if they choose to work in business, it's also the good decision. So this is our social net contacts, and please do not hesitate to ask any question, to write me and ask any question if you have it. Thank you very much for your attention. I finished my short introduction. Dear participants, any question to me? No question. I see something in a chat. Just a moment. Okay. I see the question in a chat about TISP honor program. Thank you. 2 weeks later, we will start Tomsk International Honors Program. It's a program where we demonstrate to participants the PBL approach, where also our lecturers deliver the topics in form of cases, and of course we will inform all the participants of today's meeting of free online winter school about the start of honors program. Thank you very much for your question. I'm sent all the information about it to your mails, and I will repeat it during the every day of our winter school. Now, if you do not have any questions, I'd like to introduce you our first lecturer. It's Neda Firoz, and some information about background of Neda. Now, she is a PhD student in the Computer Science and Mathematics Department of National Research Tomsk State University here in Tomsk in Russia. She graduated from some Higginbotham University of Agriculture, Technology and Science in India with a master in technology in bioinformatics, and completed engineering bachelor's degree from sorry, Nida, UPTU India University.
Okay, this is a technical university in India,
Okay.
Uttar Pradesh Technical University.
Thank you. In informational technology, she has already had teaching experience. Sorry, it's a bit noisy. 5 years in Indian University for masters and graduate students of computer application, and she specialized in computer algorithms, databases, and artificial intelligence. And of course, Neda Firoz is a lecturer and a tutor at our Tomsk International Science Program, and she delivers courses applied computer science and programming. Am I right? Did I?
Yes.
Thank you very much. Dear participants, sorry, please switch off microphones if you do not have question right now. Do not know how to do it manually. Thank you. So Nida, now it's your turn. Please, it's your time. Dear participants, Nida will deliver some lecture. The topic of lecture is bioinformatics. It's her specialization in Tomsk State University and also in Tomsk International Science Program. So I am stop talking and begin to enjoy lecture.
Okay, thank you so much, Gulnara, for a nice introduction and explaining us about the course TOMS International Science Program, which is really very important for our students of this generation. Good evening, everyone, dear participants. My name is Nida, and I will be delivering a lecture today about bioinformatics. So if you guys don't mind, you can turn on your cameras if you are comfortable with, so we can interact with each other and speak about the bioinformatics discussion. It will be healthy if we can see each other. Okay, so I will start sharing my screen until, and please do let me know if you can see and hear me clearly. Okay, great. So the first question for all of you so that you can understand what is bioinformatics and how technology is changing every single day. So anybody has an idea about what is bioinformatics? Can tell about the opinion in the chat, or maybe you can tell by your microphone. Do you know about bioinformatics? Have you heard about it? Okay. Okay, so let's get introduced to bioinformatics.
Bioinformatics is a field to understand the biological data using the software tools, etc.
Okay, okay, thank you so much. And anybody else has any idea, wants to say something? Okay, I think Junaid Swati added a nice comment on what is bioinformatics. So let's get started to introduce ourselves with bioinformatics. As you all know, the science is changing, the technology is changing every single day, and we get so many applications, so many new mobile phones coming up with so many nice technologies. So I think it's all thanks to science and the world that is evolving with the help of science. So as Edward Teller said, the science of today is the technology of tomorrow. So we need to study science, we need to understand science, and we need to improve our scientific informations, our knowledge about science, so that we can also participate in modern technology. But we should take care of our ecology too. As Alan M. Edison added, modern technology owes ecology and apology. So in TISP program, we also learn about science, technology, and also about ecology. So adding these statements, I think we should remember these statements while using technology. Let's get started with the first definition of what is bioinformatics. So as it's mentioned on the screen, bioinformatics, it is the study of computers, information, and information flow using the biological systems. I hope everybody can see my slide and what I'm drawing here. It's biological systems. You must have studied the basic biology. The basic biology like the human digestive system, the brain, and the lungs, or probably any other part of the body. So basic biology, when included with the information and information flow, especially using the computational methods of finding and exploring the genes, the genomics and proteomics, refers to bioinformatics. It is described by Oxford English Dictionary. This is the first definition. Another definition of bioinformatics is the mathematical, statistical computing methods that aim to solve biological problems using the DNA, amino acid sequences, and related information. I'm sure you must know about DNA, a little information about DNA. If you don't, then we will explore more about DNA, RNA, and amino acids today in our lecture. Okay, so I hope everybody follows until here what is bioinformatics. You can show me a thumbs up sign with the help of your emoji section in the Zoom chat, or maybe you can just add some information if you want to by speaking with the microphone. Okay, thank you. I just see one thumbs up. What about other participants? Can you really understand? Okay, another thumbs up. Thank you so much. Okay, so let's continue our discussion. So the next slide. So here we go for the next slide. And another definition of bioinformatics is coming up next. Please do tell me if you can see. I'm so sorry, I think the slide is not moving. I have to just restart again. Okay, so here is another information about bioinformatics, which is by Richard Durbin. Richard Durbin says that he doesn't think biological computing is bioinformatics. He has another idea about biological computation. He says is not bioinformatics. So he doesn't believe and he doesn't want to contribute the mathematical concepts in the bioinformatics to be the part of bioinformatics. And he says that even when connected with biology, even though there are mathematical computations in biology as well, yet the related problems cannot be solved solely by mathematical approach. So we need some sort of management and subsequent use of biological information, genetic information added towards in form of databases, in form of collection houses where we can store them and manage them and process them. So he says, if you talk about including maths in biology, and its computation, it's not the correct definition. According to him, only the management, the processing, the storing of the information at one particular location is bioinformatics. So this is another perspective of bioinformatics where we store the data information in terms of databases. And that's how he defines bioinformatics. Okay, so let's do some quick revision of certain terminologies, certain types of terms that you should know. So we will start with the beginning of cell information and machinery. As we all know that the cells are the basic part of life, you must have known about cell in your basic biology class. Cells, they store all the information to replicate itself. When we talk about human genome, it is about 3 billion base pair long. Almost every cell in human body contains same set of genes. So these are very important points related to cells. And if you heard about Human Genome Project, then you would know that today we will mark around 20 years of celebration of completing the human genome and its whole information in form of a database. It was done in 2003, in the year 2003 and the month of April. So almost 20 years to this collection of information of human genome. Then this whole cell information and machinery gives us the idea of from where the human being evolved, how the human life came on Earth. So we are able to study with the help of bioinformatics, exploring the cell into its basic components called genes, genomes, proteomics, etc. Now, there's one important point here that says not all genes are used or expressed by those cells. So we need to find which genes define the evolution of life, and then we can make the evolution tree, which you must have studied in your high school. Then there are certain machineries to do this kind of job. These machineries basically are a part of computer science, which we use softwares where we store information, collect it, and process it. So cell information and machinery is basically about the collection and storage of the data that is very, very important to human life, that is cell, the basic part of life on the Earth's surface. So if you followed until here, let's go to the next slide. Now, you must have also studied about prokaryotes and eukaryotes. I'm sure this is a very basic term that we use in biology. So I hope everybody knows about prokaryotes and eukaryotes. Any ideas? What are these kind of species, prokaryotes and eukaryotes? Anybody who wants to answer? can unmute and answer. Just the basic biology terms.
I think prokaryotes are those cells which lacks nucleus. I don't know,
Okay.
I'm not sure, but
Yes, yes.
and
Oh.
eukaryotes have nucleus. I don't know.
Yes, yes, absolutely right. So yeah, the prokaryotes lack nuclei and eukaryotes have the nucleus, and that's how they survive and they propagate their species. So according to the most recent evidence, there are basically 3 branches to the tree of life. The tree of life means the evolution tree from where the life evolved on the earth. So if we are interested in learning about how the life evolved on the earth's surface, we are definitely interested in learning about cells, and cells contains nuclei. Some species have nuclei and some do not. Those who have nuclei are called prokaryotes, and those who do not have nucleoids or nucleus in general are called eukaryotic species. So prokaryotes include archaea, these are the ancient ones, and the bacteria, and eukaryotes are from the kingdom of eukarya, and it includes plants, animals, algae, etc. So this is just about the basic study of terminologies in bioinformatics that we are going to use. And as you can see also in the picture just beside it, having some sort of tree structure, which is leading to some sort of species, starting from stamen profiles, plantae, animals, and fungi. So these are all the groups of life that are drawn in the form of tree structure. Okay, so if this point is clear, I think it's very, very basic biology, so everybody can understand and maybe revise it as we go further to the next slide. So now let's also quickly recapitulate some basic terminology that we will use today in our lecture. I'm sure you all have learned about it in your high school. So why I keep saying this? Because I really wanted to just recapitulate these terms. If you forget, let's just revisit them quickly. Genome is an organism's genetic material. It comes from DNA. It is the basic, very, very smallest part of DNA, you can say. Then the basic term that we keep using is gene. It's a discrete unit of hereditary information, which is located on the chromosome and consists of DNA. So DNA is the basic part from which we extract the gene, and out of it, we extract the genome. Then we have the genotype, the genetic makeup of an organism. This describes the type of which organism belongs to. So there are different types of genomes, and in that genomes, we categorize genotypes to classify the different organisms, for example, plants, fungi, or animals, etc. Now let's talk about a little bit about phenotype. So phenotype is the physical characteristics of an organism. For example, how tall a human being is, or for example, what is the height of the giraffe or the tree. So these are all expressed physical characteristics called as phenotypes, which will be marked and listed in a document format. Also, let's talk about nucleic acid. It is a biological molecule containing RNA and DNA both together, and it allows the organisms to reproduce. So that's how an organism reproduces. A parent genome gets transferred to the child genome, including the information that is there in the nucleic acid. If you guys understand until here, we can move on, and please keep showing me the thumbs up. In case of questions, you can keep your microphone on mute and ask questions if you have any. Okay, thank you very much. I just see 1 thumbs up. What about other participants? Okay, 2. Okay. Okay, thank you so much. So let's continue to the next section. So in the next slide, we continue to explore some more terms. Why I'm trying to introduce you to these terms, because we will be using these terms for understanding more about bioinformatics. Okay, So the genome is, as you know, is a basic part of a very smallest part of gene. It is an organism's complete set of DNA. So from DNA, we extract a genome. For example, in a bacteria, there are 600,000 DNA base pairs. In humans and mouse, the genomes have some around 3 billion base pairs. What are these base pairs we're talking about? We will talk about these base pairs in the coming slides very soon. Then also, if we learn some information on human genome, there are 24 different chromosomes. Each chromosome contains different genes, and each gene contains some sort of information, some sort of physical or some sort of functional unit of information, which is transferred from the parent to the child. There are specific sequences of DNA bases that encode instructions on how to make proteins. And you know that if there are no proteins in the body, the life cannot exist. So protein is also one of the basic parts, including the DNA, the nucleic acid, that is the RNA, and the DNA, including these two terms and the proteins. They are the basic parts of human life that allow us to function nicely. What are proteins? Let's talk about proteins a little bit. They are the cellular structure, the makeup or the component out of which human complex body is made up of. There are complex molecules, some very complex molecules included inside proteins called amino acids. Amino acids are chemicals that are part of proteins and lead to the proper functioning of human life or life in general. So now we know some different terms in biology. Let's continue our study to bioinformatics. So we decided to keep our study limited to DNA, RNA, and proteins. Why? Because these form the basis of bioinformatics, the DNAs, the RNAs, and the proteins. Can anybody just tell me the full form of DNA, RNA, if you remember from your high school learning? If somebody knows, can write in the chat or just unmute yourself and can tell me the full forms if you remember. Is there anyone who remembers the DNA and RNA full form? Don't be shy, don't worry if you don't know. You can just say yes or a no by using your microphone. Okay, I guess nobody knows and nobody remembers. Okay, so because I can't see in the chat as well. Okay, so I'll just write it for you. DNA full form is deoxyribonucleic acid. And I told you ribonucleic acid is a part of nucleic acid, right? And RNA is just ribonucleic acid. These are different types of nucleic acids found inside our body. And the information that DNA, that is deoxyribonucleic acid, holds is about how the cell is going to function. So all the function information is stored inside the DNA. And RNA, they store the information of different parts of cell, which one to transfer from where to where. Like, for example, mobile network transfers information from one point to another. Similarly, inside human body, RNAs transfer information from one point to another using certain synthesis of protein, provide templates, and also helps to digest the food. And then we talk about proteins a little bit. These are enzymes. They actually create a kind of acid or enzymes, a natural acid, which will send the signals to other cells and regulate the gene activity. That means proteins are responsible for regulation of DNA and RNA. DNA plus RNA forms the nucleic acid, and protein is the boss. Protein will control the gene activity, how it will transfer the information, and how it will function inside our body. Where are proteins found? They can be found in major part of our body, for example, hair, skin, etc. So I hope it's clear until here, all the basic terminologies to everybody. Any confusion, any questions until here? If no confusion or no questions, please show me the thumbs up. Okay, thank you. So let's continue our discussion to the next slide. Now, a little bit more about DNAs. I chose DNA's discussion and RNA's discussion because it's very, very essential, and you will know what I'm talking about now. So the structure of DNA is a double helical structure. I hope you remember this structure. I'm not good at drawing, but there's a picture besides this drawing, and it shows the structure, the double helical structure of the DNA. There are certain codes written here called A, G, T, and C, as you can see. The structure of these A, G, T, and C molecules is defined here. Adenine, guanine, thymine, and cytosine, they are ATCG complementary strands connected to each other. So basically, A is connected to T, and C is connected to G. What are A, T, C, and G? They are kind of parts of DNA, you can say, or the components which make up the whole DNA. So A is called adenine, G is called guanine, and T is thymine, and C is cytosine. A is connected or making connections or bonds with T. C makes bonds with G, and that's how it's connected. So you can see there are different colors, yellow and blue colors. Yellow and orange colors show A and T connected together. So you see here, they are connected together, making bonds. And G and C are in dark blue and light blue colors, making bonds like these. So that's how DNA looks like. So this is the basic structure. And the strands that connect, these strands that they connect, the A and T and C and G molecules are called parental strands. The strands, besides connecting from above and below layer, there are two huge layers. These ones are called parental strands, and the below layers that connect to A and T and C and G are called daughter strands, which contain heredity information passed on from the parental strands. Now, DNA, as you can see, has a double helical structure, is basically composed of sugar. I hope you know what is sugar, the glucose molecule, and also the phosphate group contains phosphorus inside it. The structure contains sugar molecule, phosphate, and also the base of A, T, C, and G molecules having their own type of acids that has been connecting inside the DNA. Okay, so until here, we now know that how a DNA looks like and what kind of components, what kind of acidic components it is made up of, what are the strands called, the parental and the daughter strand. The parental strand contains the information of heredity, it passes it on to the daughter strands with the help of A, T, C, and G molecules. So if it's clear until here, let's continue our discussion a little more about A, T, C, and G molecules. So adenosine, thymine, cytosine, and guanine, these are the four terms that you should always remember while studying bioinformatics or biology, because this forms the basis of life in any species. We represent DNA as a sequence of letters, as I told you, A, T, C, and G, and always remember the helix that we showed you before in the slide. This double helical structure is containing a bond with A and T and C and G. So you don't forget this. That's why we have just a little bit more elaboration on A, T, and C and G molecules. Continuing our discussion now, we have a little activity to do. I hope you guys enjoy this activity. So let's recapitulate the terms that we have learned. There are, I guess, 11 participants, and I'm going to share the link of this document file. Excuse me for this. here is a document file that I'm going to share it with you. Now let's do some activity to just get some fresh memory of all the terms that we have just learned. So I hope you all can see the chat. Please open this document and write your name. In front of your name, write one definition that you learned today. Any term that you remember, like DNA, gene, or prokaryotes, eukaryotes, anything that you learned. And I think everybody is able to open the document that I've attached in the chat. If everybody is able to open the document, please show me the thumbs up. If there's any problem in opening the document, please do let me know. Let's quickly do some quick activity, fun activity. If you find some information about gene on the internet, you can just paste it, or maybe if you remember what I told you, you can write in your own words in this document. Let's do this activity within the next 5 minutes, and then we will continue our learning about bioinformatics. Okay. Let's see if you guys remember what we learned until here. There are terms already mentioned in this document. You can write the definition that you understand, or maybe you can paste a picture in the document as you feel free. How will I know? Okay, yeah, someone is typing. Great. Let's get it done by every participant. When you write a definition of a term, please don't forget to write your name. It will take just 5 minutes to write, I think so. And also write your name so that I come to know who has given what definition. Just in case if you're not able to access the document, you can write your answer in the chat if you would feel like. If you think you cannot remember exactly what I told you, so maybe you can use internet, find some pictures about it and paste it in the document. Okay, I can see Junaid Swati writing one of the definitions. Great, thank you so much, Junaid. Let's also encourage other participants to type information on RNA maybe, or maybe eukaryotes and prokaryotes. If you guys have difficulty in writing in English, I suggest you can find on the internet some pictures and paste in front of it and write your name. And once everybody is done, can show me the thumbs up. Okay, there is something in the chat. You can't open, okay. Thank you so much. Okay, no problem. I'll do it on your behalf. I will write your definition here in the document. Thank you so much, Yuan. So here's this information from Yuan. I can see someone writing lacks a nucleus, have a nucleus. Please write your name also. In the document. Okay, I will share the document file here. Okay, so as you can see, there are a lot of information on this document file. Swati has written already. Garcia has written the name. Good. Okay, Junaid has written once again, thank you. And Garcia is typing about it. So Yuan, Junaid, and Garcia have written the definitions. Anyone else would like to add about genomics and proteomics, or maybe just the genotype and the phenotype? We just learned about them. Okay. What do you think, guys? Is it all that you wanted to share? That's it? Nothing else remains? Okay, we see some more information. Okay, thank you. Thank you for writing the definitions of genotype. And similarly, maybe you can write about phenotype. Okay, I think you all have done a great job. Thank you so much. I will continue with the presentation now. Once again, let's go back to our main discussion. So here is our presentation regarding the bioinformatics. So well done. Great job, everybody. Thank you so much for sharing your thoughts about the terms that we just discussed. Now let's continue our discussion. So you can see a huge picture here on your screen. I hope everybody can see my screen now. I've restarted my screen share of the presentation. Just show me the thumbs. Okay, that's great. Thank you. So you can see there is a huge picture, and on the picture there's this term called transcription and translation. Now that you already know about the term called RNA and DNA, let's just discuss the concept of transcription and translation. So here you see this is the double helical structure of the DNA. The DNA has some information in the form of AT and CG bondings. This information is transferred to RNA. This is called transcription. When DNA transfers some information to RNA, this whole process is called transcription. I hope you understand. The transfer of information from DNA to RNA. This whole process is called transcription. While when the RNA uses the information that has been transferred to process the proteins, it's called translation. Okay? Any confusion? When we transfer the information from DNA to RNA, this process is called transcription. I hope you can see that's going on in double helical structure here. And there are nucleotides connected together. And that is being transferred to DNA, from DNA to RNA. And when RNA uses this is called the translation process. When uses this for forming new proteins or synthesizing the proteins, or maybe allowing the information to be passed from the parent to the daughter strands. This whole process of synthesizing of proteins is taken or done by RNA. And this process is called translation. You can see amino acid and these binds are growing to form the protein chain, and then a lot of synthesis is taking place. So this whole process here you see is a part of translation. And the process that you see above here is the part of transfer of information from DNA to RNA. Okay, I hope this is a very simple concept of transcription and translation. Let's continue our discussion to the next slide. If there are questions, please don't hesitate to ask. If you do not understand any term. Okay, let's continue to the next slide then. Now, we have a very important term, unrevisited, or maybe we have not defined it very, amino acids. Let's understand what are these. So we talked about certain terms called A, T, C, and G. Here you can see A, T, C, G, these are the molecules that are bounded or having some kind of coding, some kind of information connected to each other. Amino acids, as described, is nothing but the chain of proteins, or you can say proteins that contains amino acids. They are connected together. Let's say amino acid 1, amino acid 2, they are connected together and they form proteins. So amino acids are just nothing but the chains that form together to make up the protein. And there are 20 different amino acids that bind together in different forms to make up different types of proteins. For example, here you can see this is one of the type of proteins mentioned, FLLVALCCRFGH. Is there a question from the participant? I can hear some music. I'm so sorry if it's, yeah, okay, thank you. So you can see here, is there a question, Fikiru? Do you want to ask something? I can hear some music. If you don't mind, can you please just put your microphone on If you don't mind, can you please mute yourself, Fikiru? I can hear some background music. I think you cannot hear us. Can you hear us, Fikiru? Hello, can you hear us? Yeah, because other participants won't hear what I'm saying then. Is it possible for you to mute, please?
Can the host mute him?
Not sure. I'm trying, but it's not possible manually from here. Okay, thank you. Okay, so yeah, we were talking about amino acids. They are some sort of chains bonding together to build up protein molecules. And as we learned that there are 20 different amino acids. So let's take an example. This is how we could store these amino acid information in form of proteins forming chains together. So you can see from the graph here on the screen. This is a graph. Just a moment, I'll clear all the drawings first. Okay, so here is a graph, as you can see, having T, C, A, and G. Again, here, this is a map, T, C, A, and G. And then T and T, if there is a match, we store it in the file in the form of T, T, T. And if there is no match, then we store it in the form of F and L molecules. Okay, and similarly, we do the mapping of different amino acids found in the proteins of different species. And then we just store it in this format. And we try to store in the computer in the form of this format, like F, L. Where there is a mismatch, we just try to store it like FLLL. And then if there is a match, then we will just continue writing the name of the strands of amino acids. That's how we prepare a file for storing it in the form of databases, or you can say text files inside our computers. So this is a kind of map that has been created with the help of amino acid proteins that connect together with each other in different species. So here is just an example how we create a file in biology. We use this file in text format, but we copy this in the text file in the computers in this way. Okay, this words, just symbols. So for anybody who doesn't know what is F, what is L, what is V, A, L, etc., if they don't know anything about these information, they will think it's just letters, simple letters, and how to explain them what these information gets translated into, we create amino acid folds. So that to them we generally call protein foldings. So these protein foldings will be in the form of three-dimensional structure, which we are just going to see them. So this is just a simple text file we store, and to make it more clear to the, let's say, the people who are doing study of amino acids, we make certain 3D structures like this. In the next slide, you can see there is the protein folding. Okay, there's some message. Okay. There is a protein folding structure from DNA. We get the bonds of AT and CG, and then they, in certain shapes or figures or connections or networks, you can say, they bind together or they connect together in every species differently. So we store the information of each species in different shapes and forms, and then we try to make a three-dimensional structure of this amino acid chain. Amino acid chain is nothing but the proteins in general. So that's why this whole 3D structure will be translated into a proper rotating structure, which is called protein foldings. And we will just visit them in the next coming slide. So as you can see, the protein folding is here, and it looks like this. I hope this is interesting for everybody to see the 3D folding structure, because it's hard to read A, T, C, G molecules, so many connections and so many things. And it's easy to visualize the 3D structure in front of you as this, instead of just reading the whole molecule structure. Okay, any questions until here? What are protein foldings? How do they form? Why do we need to make 3D structure? I hope it's clear now. So we need 3D prints of these protein foldings just to understand which molecule connects to which molecule, and it's more easy to visualize instead of writing just the connections of A, T, C, G molecule. Please show me a thumbs up if you have no questions until here. And if you want to ask any questions, please unmute yourself and do not hesitate to ask anything that you feel like. Okay, Yuan says, yeah, thank you. Anyone else has any question? Okay, Jeanette, thank you. Garcia also says, okay. So most of you have no questions. I think we should proceed to our, okay. Now we have a little bit more information about protein structure. So you can just visualize the primary structure, which is the sequence of amino acids. We just saw the 3D folding structure. Now we have different types of structures, for example, primary structure, secondary structure, and then tertiary structure. So there are 3 different levels of this 3D structure. In 3D structure, we have primary, and then we have secondary, and then tertiary. These are nothing but the refined formats of the 3D structure that has been obtained. So the first level of refinement is the primary level. Then the second level of refinement is called the tertiary level. And the third level is called the, second level is called the secondary level, and the third level is called the tertiary level. So in each structure, I hope you will, everybody can see here, this is the primary structure, just a simple structure. And then it gets converted to the secondary structure, connecting with the double helical structure of the DNA and making it a beta pleated sheet like this, and then alpha helix like this. And they both, when they are translated to form 3D perfect structure, the third layer is called the tertiary structure. And after getting the tertiary structure, if we want to do more refinedness or something, some information is missing and we want to add some information into this molecule, then we get another structure called quaternary structure. Otherwise, we just end up until tertiary structure. So this is the structure that you saw in the previous slide, folding together. And the quaternary structure is, if it is needed, then we go to the fourth layer. Otherwise, we end up until the third layer. So you can see the quaternary structure is complex, and it is very hard to read quaternary structure from basic knowledge. If you know about proteins and genes in depth, then you can read it from the quaternary structure. Otherwise, you just can read out from the tertiary structure, even though you have a little, let's say, less information, you will still be able to read it from the tertiary structure because it is less complex than quaternary structure. Okay, so this is all about protein foldings. Now we will continue to the next slide. Why are we studying bioinformatics? The very basic question that arises after learning the basic terms of bioinformatics. So I think this question must be there in your mind. Why are we learning about bioinformatics today? What's the need of learning the basic biology once again? Why are we revisiting the whole concept once again? So there are three reasons that I will give you. One of the reasons is, first of all, there are so many researches going on in this field, and you have a lot of opportunities in terms of careers. So if you have interest in biology and a little bit of information technology, or maybe computer science, a little bit of interest, then you have a lot of scope in this field, because there are definitely different types of research areas, one of them being the databases, the storehouses, or the place where we keep information of these biological data. And we have the first research field called in terms of databases. One of the databases is NCBI. I will try to show you today how NCBI works. And then the next research area that comes here is the development of tools. So those of you who are interested in developing programming languages, or maybe developing mobile applications, they may be interested in developing some applications related to biological tools that are efficient to analyze biological data. So if you have a little bit of interest in biology and more interest in computer science, you can still become a bioinformatician and make some applications for biological data. Another area that is very hot these days is about data analysis and data analysis of experimental results. So this is one of the most, you must have heard about data analysis everywhere in the advertisements. If you see YouTube videos, you see a lot of advertisements on data analysis. You see a lot of scope coming up in artificial intelligence. So yes, in terms of bioinformatics also, if we include the power of biological data, experimental results, and analysis, including with the data analysis algorithms, I think we have a nice scope in this field as well. So if you study at TISP with us, we will try to explore how is your interest and which portion you would like to be an expert in. Okay, now we move on to the next section. Again, the same question, why are we studying bioinformatics? Why is it important for me? Why can I not just study biology? Why is information technology related to biology? So as you know, there are different measurement techniques. There are different things that we can do only through computers, and we cannot do it with the manual level. So we need to combine the power of biology with computer science. So if you combine the power of computers, computation, management, storage, everything with biological data, then we end up in having nice results. Especially the best example could be the vaccine that was 2 years ago. We were struggling for the vaccine of COVID-19, but as you can see, there are so many vaccines available these days. And it was not possible except by combining the power of biological data with computer science. To develop a vaccine, to develop a drug, it takes a lot of time. It takes a lot of time, maybe with the traditional method, it takes around 15 years for a medicine or a drug or a vaccine to come out in the market. But as you can see, with the help of power of biology and the tools in computer science, we can just combine them and we can find vaccines and medicines for different diseases. I have some section prepared for you for this as well, but let's just discuss about why are we studying bioinformatics overall. So the 1000 Genome Project Consortium Nature, this is one of the most successful projects of bioinformatics, which was completed in 2003. As I told you in the beginning of the lecture, we were able to complete all the genomes in just a period of time, started from the 1990s. The work started in 1990 and it ended in 2003. And we have all the genomes of humans. So this is one of the successful projects of bioinformatics. And there is also a paradigm shift in biology that is utilizing bioinformatics in research, especially in finding medicines for diseases which do not exist. For example, HIV, we are still trying to find all kind of available drugs and combination of drugs with the help of bioinformatics. And we hope that we will be able to find some kind of vaccine for HIV virus as well. So that's why we need to study bioinformatics. I hope this convinces all of you to come to learn bioinformatics if you are a little bit interested in medicine. Okay, so let's continue to the next section. Here we will understand what is biological sequence analysis. You just saw the combination AT and CG. This is also depicted here in this figure. You can see CNG, the light blue and the dark blue molecule, AT, the this orange colored, and this molecule, this very lightly shaded orange and green, or somehow connected together, is a AT and CG molecule description in this picture. So if we take AT-CG molecule and we, for example, let's suppose keep the genome of all the species together. For example, humans and cats, they have different genomes, and we just list them together and try to find some similarities in them. Is it possible to find the similarity manually? I'm sure no. It's not possible, because there are millions and millions of genomes in humans and similarly billions of genomes in cats. So if we try to do it with numbers, with pen and paper, it's very tough, and I am not sure how much time it will take. Some say that it can take around 204 years just to read the human genome. So technically it's not possible with simple pen and paper. That's why we designed biological sequence analysis. We collect the information from different species, their genomic information, like the sequence of ATCG. As you saw the protein folding structure, you saw the file called FLALS, something like that, some information. So these information is stored inside the computer. And then we start comparing it and try to find the match of humans and other species. This helps in two things. The first thing it helps us is to find out if there is any disease which is similar in humans and other species, and we can find a medicine. For example, rats are the favorite animals for scientists to find some sort of medicine, and they do their experiments, especially on rats, to see if there is any side effect of the medicine on the rats or not. So we arrange a sequence, and in biological sequences, we do sequencing in using different algorithms. Now, these are algorithms coming from where? Of course, from computer science. And algorithms are of different types. For example, sorting algorithms, searching algorithms, what we want to do. So what we want to do is search the similarities between, let's say, human and cats. So we will use the algorithms from computers, and we try to implement in biological sequences and try to find the match or the similarities between the humans and other species, so as to provide the end result. For example, we are searching for some similarities in terms of diseases or medicines, etc. Okay, so I hope it's clear to everybody what is biological sequence analysis. Any questions in biological sequence analysis? If I'm too fast or too loud, please do tell me. I will slow down a little bit. But the main point is you should understand what it means. Okay, just show me a thumbs up, guys, so I can understand you are all following with what I'm saying. Okay, thank you. Perfect. So let's continue to our next slide. There are certain scientific information that's mentioned here on the screen. So this lists about the different scientific methods available in bioinformatics. Is there any scientific method available already? Yes, there are so many scientific informations available. It's not a science, or overall, it's a combination of two different sciences, the biological sciences and information technology, or you can say the computer sciences. If we combine the power of these two terms together, we get another field called powerful field of bioinformatics. And as we discussed the definition of bioinformatics in the beginning, it is not only the computational biology, it is also the management of the data using the databases, using the storehouses for example, clouds for our information in biology. Now, if somebody is interested in research, they can do research in bioinformatics as well. There are two different basic research areas. One area is to create the tools. For example, the algorithms. These are called the tools with which we will use to search our data. And another field is to develop the bioinformatics tool to exactly match the mathematical method. For example, any mathematical method which is used to find the similarity. So one is to build the tool, another one is to find the tool or the appropriate model or the mathematical concept through which we will be finding the similarity between the genes or doing some computational activity with the genes. So these are the most influential research areas available in bioinformatics. And this is one of the most youngest field that is emerging. So I hope you will be able to find some interest and do some sort of research in the near future in this topic. Okay, so bioinformatics is combining the power of computers in biology. Let's do a small activity. I have been talking a lot about databases. Let's visit a small database called NCBI with the help of this link. I'll try to show you how this works. I'll share this link with all of you in the chat. And I want everybody, sorry. Just a moment. Yeah, I want everybody to just open the link and go through this and see what the arrows take you through. So, oh, yes. There's a question for me in the chat box. Bioinformatics has a strong mathematical component. Yes, bioinformatics has a strong mathematical component. Why? Because how are we going to decide which algorithm is very beneficial for us for finding the sequences? For example, how are we going to implement those search sequences, etc.? So these models are derived from mathematical concepts. So biology plus mathematics plus computer science is overall bioinformatics, or you can say computational biology. Okay, guys, I want you to open this link. I will open the link here as well on my laptop, and I will share it with all of you. Just a moment, please. Okay, so I hope everyone can see my screen now. This is the page where I want you to follow. This is a small page coming up from NCBI database. This is a base, a National Center for Biotechnology Information database, which contains information about biological data and different already been sequentialized and stored in the form of different files. I hope everyone can see this. Okay, so let's see where this arrow takes us to. This green arrow, you can click here. It takes us to the main database algorithm. I was talking about algorithms from computer science. One of the algorithms is called Basic Local Alignment Search Tool. This is the pre-made or defined tool through which we will search so much from the data. So for example, there is a file and so much data is stored already in this. And you don't know where your data is located. And you want to find, for example, this data in the center. You don't know how to reach until here. So Basic Local Alignment Search Tool helps you to exactly find the data from a mixture of a lot of huge data collection. In this, we have next segment, we have to click here. That is Nucleotide Blast. The name of our algorithm is Blast. And then you can see there is a huge sequence written here, ATCG. I hope everyone can see the sequence. Please show me the thumbs up. Okay. Okay, so this sequence has already been derived from the species. And now we are interested in searching one of the local species from this algorithm. So we are using which algorithm? BLAST algorithm, Basic Local Alignment Search Tool. We click here on the BLAST, and then if you set up on NCBI, this is not the website of NCBI, you just have to go onto the site of NCBI, and then you have to click here. And then you see different, here you can summarize your search settings, what you want to search. From here, you can write down the name of the database. If you remember, if you don't remember, then you just write the sequence that you remember that you are searching for. The same way you search your file in your computer. It is a little different because you have to search a genome, not a computer file. So you have to type the whole genome. If you remember, if you don't remember, you have to type the name of the database, for example, and you can view your results here. If you are not interested in these results, then you can also view your results in the next format. The format has been prescribed here. And then here you can write your length of your query sequence, and these tabs provide the views of the results. So in whatever view you are interested, for example, you're interested in alignments, taxonomy, or the graphical summary, you can click here and see. So let's click here and find out our graphical summary. This is one of the summary that is coming out of our search. So we searched a certain sequence, ATCG. We already by default put it in the search statement. And then we find this graph. So this graph has been described for you already. And these thick red color bars, they represent the sequence that has been used to search the database. The scale here that is used here is 1, 400, 800, and 1200. This shows the residue position, the nucleotide position, that is 1 after 400. So that's how they are positioning your nucleotide. The colors that you can see here, they show the score. The different colors, the black, blue, green, purple, and the red color, they show you the scores, the matching scores, how much match has been found. So overall, BLAST score that are more than 200 are red. That means this much match has been found between the genome that you are searching and the data set that is already available in the NCBI database. So the fact that you are searching from the big information cloud, that portion which is matching is more than 200 or equal to 200. And that's how we do a small search. Then let's continue to the description tab. Here we see the different information in the form of description. So you can see the scores, the total score, the query, etc. These are all you can say information about your data that you were searching for. If you're not interested in this information, you can still just read them. Maybe you find some relevant information from here. The BLAST is the name of the algorithm that was used to search the sequence that you are searching for. Now, from this page, we move to the next page, which is called the e-value. We now learn the e-value from this section. It is the expected value, as it is described here. It is the expected value, which is equal to the number of matching sequences that you would expect to find if you search a database from the random sequences. So like you can see here, the e-value has been described in a certain way. E-value is close to 0. The program rounds to closer to 0. That means now no match has been found. Here, in this example, the e-value is equals to 1 into 10 to the power minus 21, which means that there is a very low correspondency. That means there's a very low matching expectancy here with your searching sequence. So that's how we do, that's all how we do share a sequence with the already available sequence, and we use the already made algorithms to search our sequences and do some sort of research work. I hope that makes this activity interesting. If you are really interested in bioinformatics, all these are simple activities that you will be doing in bioinformatics if you become a bioinformatician, and you will learn more about it. Okay, let's continue to our presentation. So we have just a few moments left before we conclude the presentation. I would like to ask, is there any question that you would want me to answer before I continue to the next segment? There are a few things that I want to share, just a little bit before closing this activity. Did you find this activity interesting? Are you guys still opening the page, trying to do some sort of BLAST research? Just show me the thumbs up if you are opening the browser and seeing your browser and seeing this activity that was just designed for you. Any questions? Okay. No questions? Okay, you can write in the chat box also if you have any questions and you have difficulty in speaking. I don't mind. Oh, is there a question? No. Okay, then I will conclude with my presentation. Just let me stop sharing the screen and we will continue to our presentation. A few more things that I wanted to share with you. Okay. Just give me a second, I have to share it once again. Can you all see my screen now? Okay. So here we ended our activity. First activity, combining the power of computers with biology. Now let's quickly discuss a few more things. What are the gadgets that you cannot live without? Just write the name of the gadgets or the technology that you cannot live without in the chat box. Let's just quickly discuss them. I hope everybody can see my screen. Answer the question, what are the gadgets that you think you cannot live without today in the modern world? Mobile phone, yes. Anybody wants to answer by microphone can also answer.
Laptops.
Sorry? Laptops? Laptops, okay. Okay. So you know that mobiles and laptops are one of the products of computer science, information technology. And you cannot skip the idea of living without information technology. That's what I wanted to emphasize. So very quickly, the next segment will take you to the drug designing, including the information technology. The power of computers with biology helps us to even design the drugs. Okay, so the question is, what is a drug? It's a kind of medicine that we take when we fall ill. Okay, so it's not the drugs that are harmful for us. It's the drug that means we use them for prevention of diseases or curing it or not allowing the body to fall ill again. And it's a chemical substance or made up of some kind of natural or you can say derived proteins or chemical that binds with the body. Then if we talk about drugs, there is a very interesting thing that we can do with the help of bioinformatics. We can and bioinformatics. It must be new to you, but I'm sure this is not a new terminology for the people who are already in the research world. So we design new drugs and we use the power of computers. The best example that I gave you was the vaccine development by different countries in a very short period of time. So why do we need drugs? The argument is very simple, because there are so many people dying from some diseases and they do not receive cure and they do not receive medicine on time. And we won't want these people to die so soon just because of a small disease, even though the medicine is available. So we want to design new drugs, and we want to design low-cost drugs for people who can afford them and live a healthy life. What are the different sources of drugs? Very quickly moving to the next slide. I'm sorry, I think I am not presenting this, just a moment. Okay, so we discussed we need drugs because of various reasons. We want people to live healthy on the earth, and we don't want people to die just because of lack of medicines. So next segment will take you to one of the important points in designing of drugs is the sources of drugs. What are the different sources available? The animal sources, the plant sources, then we have the organic, inorganic, and the synthetic sources available for our drug designing. Usually when we design a drug, it takes a lot of time, especially in the designing of new drugs, because we don't know the information. We have to search the information. We have to find a lot of information about that chemical before concluding that this is available as a medicine. So one of the ways that we can design drugs is by accident, which is called serendipity. That is just by luck. If we were researching in a lab, we just found some chemical compound, which is good, and we find it by luck. That's called serendipity. With luck, we rarely will find the best medicine. So we need more combined and frequent effort to research about finding a new drugs. Do you remember any new drug discovery lately? Anybody wants to write in the chat box? Any discovery that you remember, except the COVID-19 vaccine? Okay. Okay, so I will just help you. There are some discoveries made by different procedures. One of the procedure is serendipity, and another procedure is called the screening. This takes a lot of time. And then we have chemical modification, and then becomes the rational drug design. That means the traditional approach to designing the drugs. Let me take you to another slide, which will list the lot of data about a simple designing of a drug. So a drug is a chemical molecule that will bind with the protein. In this section, there is a disease. And this molecule just goes and covers up this section, eliminating that disease. So this protein molecule is inside the body of humans, And when we take this molecule, it goes and covers up that area and cures the disease. That's how the drugs work. This is the traditional example. This is a protein. And then here you see in this section, the disease has been cured by taking the appropriate small molecule called the drugs or the medicine. Then I wanted to emphasize a little bit about chemoinformatics and bioinformatics. I have been talking a lot about bioinformatics, but there is also a term called chemoinformatics, which deals with chemistry and information technology. This is usually a very small segment, or you can say the small part of bioinformatics, where we will try to find out the chemical molecules from our database using computer sciences, using, of course, not manual labor, but using the computers to search for us. So there is a little contrast between chemoinformatics and bioinformatics. Usually people get confused with these terms. I want you to remember that chemoinformatics is a separate part, and it's a part of bioinformatics. Biology and chemistry combined together with the power of computers. Okay, then there are certain Nobel Prize winners whose discoveries helped us to find out about some medicines. They are listed here. If you want, I can share this presentation with you. You can read them and just satisfy your hunger for the knowledge of these Nobel Prize winners. I really appreciate these Nobel laureates because of them we have been able to find a lot of medicines and a lot of cures for different diseases. Then here is an example of different molecular targets in chemistry that helps to produce the medicine or the whole medicine industry. And in the near future, it is expected that around 5,000 to 10,000 new targets will be found with the help of chemoinformatics and bioinformatics. I'm just scrolling through very quickly because this just represents the whole idea of designing a drug. Now we have a little bit information about what are the different methodologies available in computer science for designing the drug. So there are two. Number one is the structure-based drug design, and number two is the ligand-based drug design. These are two different terms when we talk in terms of designing. We are actually talking about the molecular structure. So one is called the direct design and another one is called indirect design. Why? Because it is followed by the spatial structure when the target is known, just like the eukaryotes and prokaryotes. You know that one has a nucleoid and another doesn't have. Similarly, here in the structure-based drug design, we know the target, then we can design. We know the place of target in the protein, then we can design the molecule to fit that target. And when we don't know the target, that's called the ligand-based drug design. So I think it's a complex term. I'm sure this will confuse you, but once you get into the drug designing concepts, you will realize these are very easy concepts and not to worry about them. Okay, and a few more minutes remaining. So let me just quickly scroll down through more important terms. Here, I will skip this activity and I will ask you to do it just after I take up all the questions because I think we don't have time. And this is not a very big activity. It will take just hardly 2 minutes or something like that. So this is the whole timeline of a drug design. As you can see in this timeline, the time that it takes is 2-3 years and 2 to 10 years. And if you scale up the whole timeline, it takes around 15 years to design a drug with the traditional. I hope I'm audible. I hope I'm audible. I'm so sorry, I can't see.
Yes, we can hear you.
Oh, okay. I'll start again sharing just a moment. Just the last part of our discussion that's going on. Okay. So here we are on this slide. Okay, I got stuck at some point and I couldn't hear anybody. Okay, I hope now everyone can hear me. So we were discussing the timeline of our drug design and discovery. So you see 2 to 5 years takes to isolate the protein. Then it takes another 2 to 5 years to find a drug which is effective against that protein. Then preclinical testing, it takes around 1 to 3 years. And then formulation of the drug, it again takes around 1 year time. And then when it gets approved by FDA, it again takes 2 to 3 years. And before that, you have to apply for clinical trials, which actually takes 10 years. So not less than 15 to 20 years, traditionally, we are able to design a drug. But the best part, using chemoinformatics and bioinformatics, we can design a drug within less than a period of 5 to 8 years. So this is a good news for people in research, and especially there are so many researchers going on for cancer medicines, as well as HIV vaccines. I hope we will be able to have some good drugs in the near future. Then we have the next slide. And with this, I think I will conclude the statement of Einstein, which says that a man who never made a mistake had never tried anything new. Even though we are making mistakes at this point of time with researchers of very different, with difficult diseases like HIV and cancer, but we hope we will be able to try and find some new solutions for such deadly diseases. With this, I think I will close this presentation and I will move on to the next section called questions. If you have questions, please do not hesitate to ask me. I am ready and open for all the questions. The activity was just about listing the drugs that you know. If you want, we can continue with the activity. The activity is just about listing the drugs, the name of the drugs like paracetamol, amoxicillin, etc. If you know them, just write down the names in the text in the chat box. And if you have any questions, please unmute yourself and please do ask questions. I will be glad to take up your questions. Maybe Junaid or Mingyu Zhao, who have been very active, like Garcia, Yuan.
We will ask about the drug development. Don't you think that there is also a dark side of this?
Dark side of
it is saving like many lives, but it is also taking many lives if you look at the side effects and the drugs, other drugs like which people take to get high, like the LSD, and so don't you think it is a serious issue?
Yes, of course, it's a serious issue and we should control the misuse of drugs. I think misusing drugs should be not normalized in different countries. It has been banned, and there are several laws to control the use and abuse of drugs. Our lecture was supposed to be finding new drugs for treating different sort of diseases. Of course, if you misuse the drugs, it is going to be life-taking and challenging for the governments to ban the misuse of drugs. Well, yes, if something has merits, it has demerits as well attached with itself. So we need to learn as a society to be able to control the abuse of drugs. And thank you, it's a very good question. I think you will be able to get the appropriate aspect in the neuroscience lecture that's coming up in the series of Winter School.
Thank you so much.
Okay, thank you so much. Any other question? Or any other points you want to say? You wanted to say something by learning bioinformatics, or you are excited about it? Okay. Okay. I don't see anything on the chat box, so that's all for me. Thank you so much for your attention. I will be glad to join all of you at the end of the winter school. Maybe we will join in the TISP bachelor's program or the honors program. We're glad to see you. If you have any questions, you can also write to at this email.
I have a question, but I don't think it's relevant to the topic of
Okay, but you can
this
ask
lecture.
in the chat if you think it's relevant to your course.
Okay, I will message.
Okay, okay. Okay, that's all. Thank you so much. I will hand over to Gulnara for the further discussion.
Okay, Nida, thank you very much for your very interesting and motivating lecture and masterclass. Dear students, I'd like to announce the program for the tomorrow. Let me share with you the schedule. So tomorrow we will have, do you see it on your screen? Yeah. Tomorrow we will have two meetings, two lectures. First of all, we will start at the same time on the same link, and we received some messages that some participants could not join because of problem maybe with the link. So we will repeat the message to all emails. So be attentive and find the right link in your mail. Tomorrow we will have the lecture on neuroscience, how to improve your memory according to neuroscience by Solomina Evgenia. And the next lecturer is Michael Semyonov. He specialized in mathematics, and he will demonstrate some interesting mathematical trick. And the topic of lecture is not boring mathematics. So we invite you to join us tomorrow and continue our free online winter school Tomsk International Science Program. Thank you very much for your attention. Take care of yourself and see you tomorrow. Bye-bye.
Thank you very much.
Goodbye.
Goodbye.